Hello, this is where I work, the Institute of Metabolic Science in Cambridge. And in it, my lab focuses on two areas. Firstly, we try and understand why some people are susceptible to obesity and others resistant. And secondly, we try and work out why, when you become obese, do you develop metabolic and other diseases. And today I'm going to talk briefly about the first and spend a little bit more time on the second, and end with something that's a rather special for our time, the intersection between, if you like, the two pandemics, the pandemic of obesity and the pandemic of COVID-19. So, in brief, obesity is, in fact, due to gene-environment interaction, and work by ourselves and others over the last 25 years have clarified the nature of some of those genes, which have helped us understand the precise biology that's going on underpinning this obesity epidemic, if you like. But very simply, when there's a famine, clearly there's never any obesity, and when food supply is very limited, it's very rare. However, in the modern era, where food is superabundant, the need to expend physical activity to get it is minimal, and it's advertised constantly, available ubiquitously, and cheaper in relative terms than it's ever been. It's unsurprising that obesity is a very common phenomenon. But the susceptibility between individuals is still highly genetically determined. And what we know now is, for example, here's a slide showing the expression of where genes are expressed, the genes that we know influence the obesity. Where are they mostly expressed? They're mostly expressed in the central nervous system. They're brain genes. And we know more than that. We can say more than just this generality. We can go right into the area of the core area of the hypothalamus. And through work in mice and in rare humans that we've contributed to ourselves, we know a lot now about the precise wiring diagram of this area of the brain, the arcuate nucleus of the hypothalamus, which is profoundly important for the control of appetite and satiety. I'll just tell you a little bit about a study that's just, it's in press now in Nature Medicine, and that we've done with the ALSPAC birth cohort, Nick Timpson and Caitlin Wade in Bristol. We took nearly 6,000 children born in a single year in 1991, as unselected as you can get in the UK. Every baby born in the hospitals, their parents were asked to provide samples, and most said yes. We sequenced the melanocortin-4 receptor in all of them and found all the mutations, and we functionally studied in a test tube every variant that we found. And here's pictures of where the variants were and the sorts of results we got when we studied them functionally. And what we found was that there was 1 in 330, and effectively, between 0.2 and those added those together, you get 0.3. So 1 in every 330 people carried a functionally compromised melanocortin-4 receptor mutation. And to just look at this complicated slide, but I want you to focus on things in red. At the age of 18, those carriers had nearly 5 standard deviations of BMI higher than those who hadn't. And the amount of weight was nearly 18 kilos, while their height at the age of 11 was also higher. So carrying these alleles made the kids tall and very heavy. And if you look at the estimate of fat mass, 15 kilograms of that was fat. So here's the picture of the individuals who carry the complete loss of function and partial loss of function mutations. Here's complete loss of function, partial loss of function for fat mass. And here are the trajectories. I think what we're seeing here in the top are just those 17 individuals who carried a single change in one gene, compared to individuals with the so-called polygenic risk score, people carrying many common variants in many genes. You can see how powerful just this one gene is in determining what sort of weight you carry into adulthood. So heterozygous mutations in MC4 are now found in 1 in 330 unselected people in the UK. We estimate, as I say, 200,000 carriers in the UK. At the age of 18, the extra weight carried is 18 kilos, 14 of which is fat. This is far bigger than was predicted by UK Biobank, because UK Biobank tends to recruit very healthy people. So we're getting a much bigger result in our unselected birth cohort. And the impact on individual care is much larger than the polygenic risk score, and we can start seeing the effect really very early in life indeed. This makes, I think, MC4R a compelling candidate for targeted therapeutics. So I'm going to move now to the adverse health consequences of chronic overnutrition. Energy intake exceeding energy expenditure is the cause of an expanded triglyceride fat mass, or what we call obesity. Obesity has many adverse effects, including of which are mechanical and gravitational. Those expansion of fat mass in the abdomen, producing reflux esophagitis, the weight producing osteoarthritis of the knees, the narrowing of the airways producing sleep apnea. But it's not immediately obvious why this expanded mass of triglycerides would lead to either cancer or indeed metabolic disease. One of the critical phenotypes that are linked to all of these metabolic abnormalities is the problem of insulin resistance. As you get fatter, fasting insulin rises to try and control blood glucose, and there's a strong relationship of that to body mass index in the general population, of course with a huge variability between individuals. How do we get from sustained positive energy balance to this defective glucose handling in liver and muscle? It's not intuitively obvious how that happens, but I think if you asked 1,000 diabetologists these days, they'd say, well, we know, doc, because we've heard all about adipose inflammation. We've heard all about the big bad fat cell, and all about these cytokines that fat tissue produces, and they must be causing the mischief in the liver and muscle. And indeed, if you only followed the animal literature, you would really be compelled by that, because it's very strong. But what I would say is, looking at human genetic and human pharmacological literature, this is not very well supported by either of those bodies of work in our species. And perhaps there's a stronger argument for a different way of looking at this problem. And that is that adipose tissue is the safe and professional place where we keep our excess nutrients. And it's only when we stop being able to safely store them in the nutrients, then we redirect those nutrients to non-professional macronutrient storage tissues. I started my own work on insulin resistance many years ago, trying to see what we could learn from studying rare humans who were very insulin resistant. And with my colleagues Anna Crook and Rob Semple and David Savage, we discovered many people who had defects in the insulin signaling pathway, resulting in severe insulin resistance. However, we were struck by a few things. We were struck by the fact that while these people shared quite a few phenotypes with individuals with typical metabolic syndrome, there were many phenotypes that they did not share. In other words, messing up insulin signaling was not sufficient to cause the low HDL, the low adiponectin, or the high liver fat we see in typical metabolic syndrome. So that led us to scratch our heads and say maybe the thought that finding out about insulin signaling would lead us to all the answers might not be the right way forward. And yet we were staring in our face, really, were individuals with the problem of lipodystrophy, and we'd been studying those for many years, myself and David Savage particularly, trying to find genes that cause people to be unable to develop fat tissue, or at least inadequate fat tissue. And of course, when we studied these people, they had all the features of the metabolic syndrome. They had everything that you would see in an obese person with type 2 diabetes, but they just had it more severe and earlier. So I'm just going to tell you a little bit about one of those disorders, perilipin 1, which is not common, but I think is a beautiful example of the sorts of thinking that you can bring to the problem by looking at a well-worked out single example. This is an autosomal dominant form of lipodystrophy, where the mutation in perilipin occurs at the end of the protein, causing an aberrant C-terminal tail. And this is inherited in a dominant fashion, and all these unfortunate people get every feature of the metabolic syndrome, even though they're not obese. They look quite muscular and fit, but in fact they have partial lipodystrophy. Why is that? How do they get all of these diseases? Well, here's the reason why. Perilipin 1 lives only in your white fat cell, and it lives only on the surface of the white fat droplet, and it has a partner molecule, CGI58. And when you're fed just after a meal, they're irrelevant. They just sit there doing nothing because your lipid droplet is busy making fat to put into it. But of course, overnight, your heart contractions are totally dependent on free fatty acids coming from your adipose tissue. So you must change that situation in the fasting state. And you do it by changing the hormones that happen after fasting. And those hormones induce phosphorylation changes on perilipin and indeed on the lipases, the enzymes that break down fat. Here on the C-terminal, for example, it is a phosphorylation site that kicks off CGI58. And so the first thing that happens in the middle of the night, and will happen to you all in the middle of the night tonight, without it you'll become very unwell, you will get ATGL coming to the surface of the droplet, breaking down triglycerides and starting to release the free fatty acids that your heart needs. Similarly, HSL, the second enzyme, will be phosphorylated. It'll be attracted to the other phosphorylated perilipin, and in a beautifully coordinated set of absolutely necessary enzymic reactions, they will deliver you the free fatty acids that your heart needs. So what goes wrong with these unfortunate people? Well, this is these people in the fed state. Instead of being like this, they're like this. That's because they lack this binding site for CGI58, so it goes around promiscuously, and all day, whether you've been fed or fasted, it's telling the fat cell droplet to break down, and it's delivering free fatty acids whether you need them or not. And they are causing the insulin resistance, the fatty liver, the atherosclerosis, and driving it through this very simple pathway. So we turned to our excellent colleagues in the MRC epidemiology unit and asked the following question. We asked for markers of the metabolic syndrome, high fasting insulin, low HDL, high triglycerides. Can we look at a large group of people, in this case nearly 200,000, and find the variants in the genome that predispose you to having a high insulin, low HDL, and high triglyceride? In this case, the insulin was adjusted for BMI. So this markers, if you like, of the metabolic syndrome. And our clever colleagues found 53 such variants in the genome. And here they are, enlisted down here. And of course, red is increased, blue is decreased, red is increased, and these are what we define. So these results are self-fulfilling. But then you start looking at these SNPs in other populations with type 2 diabetes and coronary disease, and say, yes, in fact, if you carry those, you are more likely to get coronary disease and type 2 diabetes. But if you look in the middle here, there's quite a lot of blue. And what is that? Well, these are measurements of body fat percentage and BMI in other populations. And somewhat surprisingly, these risky SNPs, these risky variants, seem to be associated with lower measurements of that. So we decided to look in a bit more detail and went into populations that had DEXA, measurements of body composition, much more detailed regional measures. And what we found there really surprised us. Because I think if you'd ask many people what is a waist-hip ratio about, it's about having a big belly, and the visceral fat is what's really important. But these risky SNPs, as we call them, the risk SNPs, are not really associated with that much change in truncal fat, a little bit of increased visceral fat. What they're really associated with is lower amounts of fat in the gynoid, the buttock area, the hips and thigh areas. And so when we looked at where those genes are working, not surprisingly, they're influencing fat. These tissues that they are being influenced by these genes are adipocytes and fat cell genes. And when we go into the laboratory and start playing with these genes in fat cells, we show that the genes have a profound effect on adipose differentiation and adipocyte function. So, in effect, I often use this analogy to help explain what we're finding to a lay audience. I call it the soggy bathroom carpet model. I know no one has a bathroom carpet. I did in my youth. We didn't have any central heating. We had a carpet in our bathroom. So if you have a tap turned on and the plug is out, you can remain healthy with a nice pristine bathroom carpet as long as you store your triglyceride or your water in your bath. Now, we normally think about obesity and metabolic disease in the following way. We think about people eating too much, having too much energy intake, or people not expending enough energy, having a narrowed output, and then you overflow and become sick with your soggy bathroom carpet. What we don't really think enough about is, does everybody have the same size bath? And of course, the answer is almost certainly no. These individuals who carry lots of those risk SNPs I told you about, those will actually make you have a smaller bath. So that for any degree of imbalance, you're much more likely to become sick at an early stage of what we would call obesity. Whereas if you had a lack of those adipose SNPs, if you like, those variants, you'd have a big bath and you could happily become obese without developing the metabolic complications of obesity. I mean, here's an example of how we might think about dealing with people with these limited size baths. We don't really have many drugs as yet that can improve your bath size, but we can much more effectively these days turn the tap down. Here's an example of a patient who attends our national clinic for people with lipodystrophy. You can see that she has very little fat in her limbs and buttocks, carries all her fat centrally. She's just moderately overweight. She's obese, but she's not massively obese, and yet she's a patient with severe diabetes on massive doses of insulin. But just a few months later, when she's had undertaken bariatric surgery in the January of that year, within a few months, she's off all insulin. Her diabetes control is completely reversed. And what we've effectively done here, by reducing food intake and maintaining it reduced through the bariatric surgery, we've taken that pressure off, turned the tap down, and allowed her very small bath, if you like, to hold this energy balance with tremendous benefits for her metabolic status. We are, of course, trying to be smarter than that and not using things as crude and brutal as bariatric surgery. Here we're using human genetics to try and identify new pathways that we might target pharmacologically. We found, for example, a rare variant in a gene called ALK7, a serine kinase expressed in adipose tissue. And people who carry this rare variant, about 2 in every 1,000 people, have a lower waist-hip ratio and a lower risk of diabetes. And when we look at this variant, it's very conserved across all the species, so it looks like it's really important. And when we look at it in the structure of the molecule, it's at a critical junction at the signaling end of this molecule. When we mess this around from an isoleucine and put a threonine in, this would really block the proper movement of this molecule within the cell. And Nuno Rocha in the lab, a really talented postdoc, created a system that allows us to study this variant. And what Nuno found is that this mutation causes a severe defect in signaling function in red here compared to the wild type or constitutively active ALK7. Now, this sounds very techy, but what it basically says is that if we developed an ALK7 inhibitor, we may allow safe expansion of fat mass, and therefore protecting against the metabolic consequences of overnutrition. So the lessons that we've learned from studying the genetics of human insulin resistance, if we have people with disorders of insulin signaling, they're not really like people with common metabolic syndrome. They only have part of the problem. If we look at people who have lipodystrophies, their problems look just like those of obese people with metabolic disease. When we look at the genetic variation that underpin metabolic syndrome in the general population, they're dominated by genes in adipose tissue. And when we look at what's going on there, it's much more consistent with the idea that having an inability to expand your adipose mass safely is really the critical determinant of metabolic health rather than the inflammatory component within adipose tissue. And now we're using large-scale population genetic studies, including now exome sequencing, to identify more precisely targets that we can manipulate for health benefits into the long term. I'd like to finish today by talking briefly about the obesity-COVID link. Is it real? It certainly is. Here's some work that I was kindly allowed to participate in by Sue Jebb and Paul Aviard at Oxford. It's just been accepted in press for the Lancet Diabetes and Endocrinology. It follows up on other UK-based studies. It's based on nearly 7 million GP records and then following people through to hospital admission, ITU admission and death. And you can see that the impact of body mass index on ITU admission really starts at around 25. So even overweight people are starting to show an increased risk of ITU admission. And it's not just obesity. For every unit increase in obesity, there's definitely a unit increase in hospitalization, ICU admission, and death. But if you take obese people who have markers of the metabolic syndrome, pre-diabetes, fatty liver, etc., they have much stronger associations. So it's not just the mass of adipose tissue itself, it's the metabolic complications of adipose tissue that seem to be really predisposing to these adverse events. How might that be happening in COVID? Well, I think we really need to look in detail at why people with COVID-19 who do badly, why do they do badly? They largely do bad because they end up in intensive care on a ventilator because their lungs can't exchange oxygen properly. And that's because they develop a profound inflammatory and thrombotic disorder in the lungs. The virus comes down, it enters this so-called type 2 alveolar pneumocyte where it proliferates grossly, causing cell death and cell fusion and an enormous inflammatory infiltrate. Very characteristic of COVID pneumonia are these complement activation and the presence of coagulation, lots of clots within the lungs. Here's a picture of a person who sadly died of COVID-19, and here's a typical COVID-19 picture of this microthrombus. This is a clot in the capillary. You basically don't see these with other types of pneumonia, or other types of lung damage. This is very characteristic of COVID-19. So how does obesity worsen COVID outcomes? A simple thing to say first is it's not, almost certainly not, because we have big bellies pressing on our diaphragms or big heavy weights on our chest, because there are lots of forms of pneumonia and they don't all do worse in people with obesity. So this obesity link seems very specific, very closely related to COVID-19. And of course, there are numerous possible ways that could happen. There are mechanisms through the insulin resistance and hyperinsulinemia, which we know causes endothelial dysfunction and indeed platelet dysfunction, increases the antifibrinolytic PIA1, therefore contributing to persistence of clots. We also find things you may not be expected. Adiponectin. Adiponectin has surprising effects on suppressing pulmonary inflammation. Adiponectin is very low in people with obesity and insulin resistance. If you put TNF alpha on pulmonary endothelial cells, you markedly increase markers that attract circulating leukocytes, and adiponectin can block that. And adiponectin levels are very low in individuals with obesity and insulin resistance. Here's complement, which, as I mentioned, complement deposition is really strong in the lungs of people with COVID-19. Complement C3 levels are increased in the obese. The good news is if you even go on a 6-week diet, you can drop your complement levels markedly and well before you lose very much weight. And that's part of, I think, the message for obesity and COVID. Losing even a small amount of weight can have these benefits on things like complement, adiponectin, and inflammatory markers before you lose much weight. And even now we're beginning to understand that the so-called ectopic deposition of lipid may not just happen in liver and muscle, but there is a phenomenon called the fatty diabetic lung, at least well established in rodents, and perhaps could this be contributing to the susceptibility of the lungs in people with obesity? So I'll finish by thanking my colleagues and collaborators and thanking you for your attention and wishing you an excellent conference.